Hello everyone, our topic is different RNAs in the same cell. Our group members are 张宏宇、陈佑宇、龚宇豪、傅冠军. Humans are composed of cells, and DNA RNA is the material of cells. But I like RNA more than DNA because it has U in it. Contents of this report. First is SDGs. Second is introduction. Third is summary. Fourth is experiment. Fifth is discussion. Sixth is reference. Our report is connected with SDGs 9, Industry, Innovation and Infrastructure. Enhance scientific research. Upgrade the technological capabilities of industrial sectors in all countries, in particular developing countries, including by 2030, encouraging innovation and substantially increasing the number of research and development workers per 1 million people and public and private research and development spending. RNA is a molecule that is present in the majority of living organisms and viruses. It is made up of nucleotides, which are ribose sugars attached to nitrogenous bases and phosphate groups. The nitrogenous bases include ethine, guanine, uracil, and cytosine. RNA mostly exists in the single-stranded form, but there are special RNA viruses that are double-stranded. The RNA molecule can have a variety of lengths and structures. An RNA virus uses RNA instead of DNA as its genetic material and can cause many human diseases. Transcription is the process of RNA formation from DNA, and translation is the process of protein synthesis from RNA. The means of RNA synthesis and the way that it functions differ between eukaryotes and prokaryotes. Specific RNA molecules also regulate gene expression and have the potential to serve as therapeutic agents in human disease.
mRNA is transcribed from DNA and contains the genetic blueprint to make proteins. tRNAs are RNA molecules that carry amino acids into the ribosome. rRNA forms ribosomes, which are the protein synthesis machines.
Today, I'm going to tell you the difference of mRNA and miRNA. mRNA structure and length are longer and more complex, but as you see, microRNA is shorter and simpler. MicroRNA is only present in eukaryotic cells, but absent in prokaryotic cells. But mRNA is present in both eukaryotic and prokaryotic cells. Between its gene and regulation, mRNA molecules are directly involved in the expression of genes by encoding the instructions for protein synthesis, but microRNA is regulate gene expression by binding to target mRNA molecules. And with its function, microRNA's function are as epigenetic responser of protein translation, while with miRNA, its functions are as a messenger, carrying messages from DNA in the nucleus to ribosomes on the ER where it is first translated into a corresponding sequence of amino acids, then synthesis into protein. As you can see, abundance and diversity between mRNA and miRNA. mRNA's abundance and diversity. Multiple mRNA molecules can be transcribed from different genes, simultaneous in a cell, but microRNA are a class of small RNA molecules, and each microRNA can target multiple mRNA molecules. In its transportation and protein production, we can see mRNA molecules are directly involved in the translation process, but miRNA molecules do not code for proteins themselves. The experiment. Our experiment is simultaneous detection of microRNA and mRNA molecules in leaf tissues of rice and maize. The experiment summary. Detecting the simultaneous presence of microRNA and mRNA in a specific tissue can provide support for a prediction that the microRNA regulates the mRNA. Also, two such methods have been developed for mammalian tissues. They have a low signal-noise ratio and poor resolution at the single-cell level. To overcome these drawbacks, we developed a method that uses sequence-specific microRNA-locked nucleic acid mRNA-LNA probes. The experimental. The rice cultivar tongue 67 was used as wild-type accession for japonica rice in this study. The rice seeds were sterilized by 2.5% sodium hypochlorite and then placed on a wet paper towel at room temperature for a week. The sprouting seeds were moved to soil and grew in a walk-in growth chamber at 28 degrees Celsius under 16-hour light and 8-hour dark cycle.
好，现在应该是暂停的阶段。好，3、2、1。
Now I have talked about the experience method, RCA. RCA is rolling circle amplification. It is a commonly used research tool in molecular biology, material science, and medicine. RCA is an isothermal enzymatic process that uses DNA or RNA polymerases to produce single-stranded DNA or RNA molecules, which are a connection in a series of complementary units of a template. And next page, I have talked about the graph. According to this graph, it shows the ZMGRF8 and its potential microRNA regular, ZMA-MIR 396F. In developing mice lives at 48 and 120 hours post-imbibition. This graph shows that at 48 hours post-imbibition signals of ZMGRF8 are detected in short apical meristems and serially developing leaves, but no signals of ZMA-MIR 396F is visible. In contrast, abundant signals of ZMA-MIR 396F are detected in developing bundle sheath within the middle layers of the first leaf, and no signal of ZMGRF8 is seen at 120 hours post-imbibition. These expression profiles are consistent with relative impressions levels determined by QRT-PCR, suggestion that our method works properly for detecting both microRNA and mRNA transcript in one's run of detection probe labeling. Relative expression levels of ZMA, MIR 396F, and ZMGRF8 after imbibition assayed by QRT-PCR. The lowest expression levels of ZMA MIR 396F occur before 60 hours, and the highest expression level occur at 120 hours post-imbibition. In contrast, ZMGRF8 shows the highest impressions level at 48 hours post-imbibition. I'll pass the microphone to Chen Youyu, who will describe discussion. Now I have talked about
The technology allows researchers to study gene expression and regulatory networks at a high-resolution level, detecting miRNA and mRNA molecules in plant tissues by combing LNA probes and padlock probes with RCA to achieve high sequence specificity and sequence-based signal amplification. The method is robust, versatile, and scalable to meet the emerging demands for detection of gene expression at the single-cell level. This is our reference. And this is our group assignment. In conclusion, RNA is an important material in our cells. There are many kinds of RNA in the same cell: rRNA, tRNA, mRNA, and miRNA. So I hope that my presentation today will help you understand more about RNA. Thank you for listening. Thank you.
Thank you.
Thank you.